Okay, let's move on to the third point about the innate immune system, which is the cellular responses. Now, I want to be very careful around here because a lot of these cells involved in the innate immune system do have a role in the adaptive immune system. So what I'm only going to be talking about is certain actions that relate to the innate immune system, not the adaptive immune system, which we will cover next week. My tea's getting cold, so hang on. I always do this. I always let my tea get really cold, and then I basically have to chug it because cold tea is disgusting. Alright. Hopefully I won't burp on stream. That would be mightily rude. Apologies. So the cells involved in the innate immune response of the body. So non-specific immune responses here. We're not talking about adaptive immunity. This is just non-specific stuff. So key players in the cellular responses are, we've already spoken about this before. We're going for the macrophage, the neutrophil, the NK cell, which stands for one of the best names in immunology, the natural killer. Natural killer cell. We also have contributory roles from cells called mast cells. And lumbered in with these as well, which I'm not really going to speak about too much, is you have things called eosinophils. INO. And basophils. But we're not going to talk about too much of those because their function is not kind of a major, I wouldn't say it was a major function of the innate immune system. When you're comparing macrophages, neutrophils. Oh, and of course we've got to think about our epithelial friends. They are not typically immune cells, but they are definitely involved in the innate immune system because they line all of your exposed sites to the outside world. So they're kind of the first responders, if you like, or the first instigators of the response. So let's put epithelium up top. Can't forget those. And we'll go through these briefly and explain kind of briefly what they do and what they are responsible for. And okay, let's start at the top. Let's start like as if you were having an infection. So let's start with the epithelium, as opposed to the barrier function, which we've already talked about. So they are obviously barriers. But they're also able to sense bacteria, viruses, fungi, parasites. And they do that. I mean, we say the word sensing. Realistically, what's actually happening is that they contain proteins within them that respond to the presence of certain bacteria or viral, or say, let's call them pathogenic, or even better, let's call it non-human structures, chemical structures and proteins. So they will have, they recognize and put that in inverted commas, pathogen. They're called molecular patterns, or we'll just call them patterns here. So these are non-self proteins, usually proteins or sugars. Or sugars on proteins if you have glycoproteins. And they do this through proteins in their own, in the epithelial cells called pattern recognition receptors. Or PRRs. PRRs. And these, oh, where have I gone? Hello. There we go. So what you might have is you'll have, say, your epithelial cell. Say let's take a lung epithelial cell with the cilia on the top. Draw a couple of these. It's like feet with loads and loads of toes. Okay, and you've got a nucleus down there. And PRRs, which we'll draw in yellow. So you might find that these PRRs are on the surface of the cell, in the membrane. They could be within the cytoplasm. Just going to very, very quickly scribble these. These are just proteins on the surface or proteins in the cytoplasm that are specific for general things that are present on viruses and bacteria that aren't present on humans. So it's a pattern record. It's just a pattern, nothing specific. It can't tell the difference between, say, an HIV virus and a flu virus. Can't tell the difference between a bacteria. It can't even tell the difference between a bacteria and a virus. It's just things that it just recognizes things that are not self. And when it does, what you get is the first step really of the immune response. So say you've got your this cell in the middle, unfortunately is going to have some bacteria or some viruses within it or on the surface of it. And these pattern recognition receptors, they're going to activate. And they're going to signal to the nucleus of the cell where all the DNA is kept, and it's going to signal to the nucleus to say, you need to make some antimicrobial proteins. So and these are proteins that will be detrimental. There'll be enzymes that would degrade certain bacterial structures, certain virus structures, that kind of thing. One of the bigger things that they do is they also trigger the cells to produce something called chemoattractants. So these are chemical, well, they're proteins, generally, that drive the recruitment of other cells. So other cells are able to sense these, and they're brought into the area. And if you ever have like an infection or a cut even on your hand, and it gets swollen up and it gets raised, after a certain amount of time, what will happen is that immune cells from the innate immune system will start to flood into the area. And they're brought there by these initial chemoattractants that are released from epithelial cells or other cells that are damaged or infected, that kind of thing. So these attract other cells. and you might often hear them referred to as chemokines. Hi folks, sorry to interrupt, but I wanted to thank you for making it this far through the video, and to let you know that I really appreciate your engagement and I hope you're enjoying the content. Everything that you're watching here was live streamed over on my Twitch channel, twitch.tv/4085. I stream 3 times a week, Monday, Wednesday and Thursday at 9 PM UK time. And if you want to get involved in any scientific discussions, ask me any questions, or just watch a bit of Xbox gameplay, you can head over there to do so. This YouTube channel is dedicated to the science content. I have a second channel where I post highlights of gaming sessions and other miscellaneous content. The link to that channel is in the video description. Thanks again for your time. Now back to the video. I should be an engineer. They attract other cells and they're very, very important. Okay, so that's your epithelial cell. It's the first line of kind of defense, the first thing that can sense, in inverted commas, sense pathogen invading. And it responds by making antimicrobial proteins, chemoattractants, and sets up a response from other cell types. One of those other cell types that comes along is the macrophage. So let's talk about macrophages. Now we've spoken a little bit about macrophages in the in this part over here, and the fact that they're able to eat up bacteria that are bound together with IgM antibodies. So they can phagocytose bacteria. Eat. So another thing that they are very, very good at, macrophages, is they are the go-between between the innate immune system and the adaptive immune system. So they actually are one of the major triggers of the adaptive immune system. And they do this through, I'm not going to go into it today because it's not about the adaptive immune system, but they do this through a process called antigen presentation. That is terribly drawn, terribly written. I'm sorry, but I'm not going to redo it. Antigen presentation is where a macrophage, or lots of other cells can do this, but macrophages do it quite readily. They take up bacteria, they digest the bacteria, and then they display a small proportion of that bacteria on effectively what is like a plinth. It's a molecule called MHC class 1. MHC class 2. It's a molecule called MHC class 2. And what that does is it triggers a subset of cells called lymphocytes, specifically T lymphocytes, to recognize that bacteria and start mounting an adaptive immune response against it. Antigen presentation on, and we'll go through this in the next week, on MHC class 2 to T cells, T lymphocytes. Okay. These cells, these macrophages, are also able to produce chemokines. As the epithelial cells were, and so they can draw in more cells and propagate the immune response. So they are the go-between, mainly the go-between between the innate and the adaptive immune system. Very, very important cell type. Right, so moving on, we have the neutrophil. Zoom in a bit so we can see this. So the neutrophil was first described to me by my old professors as the hand grenade of the immune system. Neutrophils are full of what are called granules. So you have a neutrophil, lovely irregular shape, nucleus, fried egg. Then the neutrophils are filled with all of these vesicles or granules or whatever you want to call them. And the granules contain quite destructive enzymes. So you have proteases, you will have antimicrobial peptides, and the like. And what these cells effectively do is they're very short-lived cells. They don't live for very long, and they probably don't live for very long because they're quite dangerous. So short-lived. And when activated, and they can be activated by a number of different factors. They can be activated by cytokines, which are proteins released by other cells. So like your epithelium will produce cytokines in response to bacterial infection. And they're also activated by complement, which we've looked at in the previous section. And also by direct contact with pathogen via PRR, which we've discussed. And the activation of a neutrophil results in something called degranulation. And this is where it releases all of those vesicles that are filled up with your proteases, your antimicrobial peptides, and all those kind of things. And effectively spewing all of that over the area that's got the bacteria or the virus or the pathogen within it. So release of granules. And this can cause destruction of bacteria, viruses. Oh, that is... I'm going to rewrite that. That looks like ulcer. Viruses, pathogens, just general pathogens. And importantly, it can also destroy tissue, human tissue. It's very nondescript. And neutrophils and the degranulation of neutrophils are responsible for a lot of the redness and the pain that is associated with, say, cutting yourself or getting an infection. It's neutrophil degranulation damaging the tissue around. It's all in an attempt to get rid of the pathogen. But they are effectively like small hand grenades. You don't really want them to be overexcited in the wrong context.